Okay, thank you all. Thanks to ChemAxon, our partner now for many years. I think it's 6 years now. It's always interesting, funny, and everything is good. Food is good, science is good, and very inspiring meeting. So thank you again. Okay. For those who don't know probably much about Aureus, what we do is develop and organize data into knowledge bases. So this is actually the chart of the Aureus process. So we extract information from patents and journals, and we extract all the pertinent information related to structure-activity relationships. So this morning there were some talks about extracting chemical structures. We don't do actually automatic extraction, so we have a team of analysts who read the publications, the patents, and extract all this chemical and biological information which are then structured into a platform, unique platform, which is actually the Horoscope knowledge basis. So these bases, for the moment, include and cover different target families. For instance, we have now covered the GPCR, kinases, ion channels, proteases, nuclear receptors, and also ADME and drug-drug interactions. Many applications have been developed to mine these databases, and here where ChemAxon has actually helped us to maintain the chemistry part and develop application, web-based application to extract these informations. So we have developed what we call the OrQuest, which allows the end user to build combined queries for searching the chemistry part, or also to use all the biological parameters available in the database to make, for example, selectivity profile or retrieve active compounds against specific target and so on. I will talk more specifically today on OR Profiler, which is one of our latest applications. And again, here we have implemented a lot of ChemAxon capabilities. Okay, like I said, we cover all these target families. So we have three main components: the chemical space, which is actually structures and all the related information on stereochemistry, salt, the pharma codes, the names, synonyms, the biological space, which is the whole information we extract from the peer-reviewed papers and patents on the biological data. So we have information on the data point, what information, biological information are behind this data point, such as target information, the biological protocol, the biological material used, and so on, and of course the endpoint activity. So far we have covered all these numbers. I'm not going to go through them. We have more than 3 million biological activities now associated to more than 800,000 unique compounds. Aureus, of course, provides other services, for example, on-demand databases. We are working actually on different thematics, immunogenicity, biologics, hepatotox or cardiotoxicity database or transporters, and so on. Of course, we provide also on-demand reports and studies, and more specifically on drug-drug interaction reports. We have been involved in many European projects, more specifically with the ChemAxon as partner in the No-Tox project, which actually ended up with developing metabolizer as a product in ChemAxon portfolio. As I have already mentioned, different applications have been developed to play with this data, or Quest, or Profiler, I will talk about now. Or Pass, which allows to predict biological activity spectra, and DDI Predict, which uses all the PK data we have in the ADME database to make drug-drug interaction predictions. So the OR Profiler application allows the end user to generate pharmacological profiling and alerts by profiling compounds or profiling targets. So you can have new insights into target selectivity, or have some hot activities on off targets, and also assess the chemical space associated with your target. Of course, all this data you can see in this heat map here are experimental values that have been extracted from journals and patents. I'm going to go very fast. These are the tools that have been integrated in OR Profiler. So the search is maintained by JChemBase. We have used the calculator to compute some molecular descriptors which can be used to filter and fine-tune your profile and screen for both chemical and pharmacophoric search. Okay, so here I have an example of chemical similarity on your compound. Again, you can search by name or draw structure and run your profiling either by exact search or similarity search. And you will have rows of molecules, and here you have all the targets that are reported on these molecules and activities here with a range of biological activity, red for very active and blue for inactive. I'm going to switch this one. So to fill these gaps here, these actually are missing values, either not published or not captured yet in the database. Most of the time not published. So we have moved to a next step to develop a module that allows to predict some of these activities by collaboration with Professor Vladimir Proikov and his team. So this is a screenshot of the results. Our future directions, of course, to deliver additional knowledge databases to extend what we call the global pharmacology space. We are more specifically working now on drug safety databases and, of course, leverage some European funded projects involving our partners and pharma players. Thank you for your attention. I'm around. If you want to play with the system and run your own queries, please contact me, and I will be happy to give you a demo. Thank you very much.